we've seen what goes on inside cells in fairly general terms, how pathways can get electrons out of molecules, and the electron transport chain can convert electrical current into another form of energy that can eventually be converted into ATP. So that all goes on inside cells. And to understand that in detail is challenging, and most of the detail is not in this course, but that's okay. What I want is to see it from outside the cell, to see what a cell would actually be thinking about, so to speak, or looking for in the environment, because this is what has a huge effect on the entire world. So let's think about it from a big picture perspective. And so the first thing we think of is respiration, and that's what we've been talking about. So for review, our electricity, you have an electron donating reaction here, electron accepting reaction here, and if you can connect them with a wire, electrons will flow, and that's a form of energy, right? So respiration is using that. It's using an electron transport chain to make a proton gradient that ultimately makes ATP. And so for this to work, there must be an electron donor that gets consumed, so something like glucose that a cell eats, so to speak, and then a terminal electron acceptor that also gets consumed, so something like oxygen. So both of these things must continually be consumed by the cell for this to keep going. And this is like our cells. They need glucose or other sources of energy, and they desperately need oxygen. If they don't get oxygen for a few minutes, they cannot make any ATP. The cells that can do fermentation can make ATP without oxygen, but you'll see it's not nearly as good. And our neurons cannot do that. So human neurons will die without oxygen because they can't maintain themselves. They can't get the ATP they need to keep their processes going and repair their membranes and their proteins. So in respiration, the flow of electrons is the key thing here. And the kind of respiration we've been talking about, there are two types. One is aerobic, one is anaerobic. The type we've been talking about is aerobic respiration. That's when oxygen is the terminal electron acceptor. So that's what human cells do. That's what lots of obligate aerobes and facultative anaerobes do. And your physiology textbook may have called this cellular respiration, and that is stupid term, because most cells on Earth use different types of respiration. Most cells don't use oxygen as their terminal electron acceptor. But if you look at animals or plants, they do use oxygen as their electron acceptors. And so they use aerobic respiration. And all of our nucleated cells do this sometime. So here is the chemistry of aerobic respiration. Again, that's this reaction. Glucose plus oxygen becomes carbon dioxide and water. These are the two half reactions that make that up with the electrons in pink. The donating reaction converts glucose to carbon dioxide and free electrons. There are also protons that we just kind of ignore. These are released in the pathways. So these are not part of the electron transport chain. These come from the chemical reactions in the pathways. But an equal number of protons are consumed by the oxygen reaction, where 24 electrons would combine with 6 oxygens to make the waters. So those are the half reactions that power glucose-based aerobic respiration. And we can think of it this way, where glucose donates electrons to electron carriers, which donate them to electron transport chain, and they go to oxygen. And that electron transport chain is a motor. Glucose, other things happen, right? It goes through those 3 pathways, and lots of other things happen, and that's what's shown here. There are waste products of that. Oxygen has a waste product, water. That's aerobic respiration. anaerobic respiration is what would be new to us, right? It's what you wouldn't have thought about if you haven't taken microbiology before. Anaerobic respiration uses anything else as the terminal electron acceptor. So anything but oxygen is anaerobic respiration, and many bacteria and archaea have evolved to do this. So a very common, I don't want to skip a slide, a very common form of anaerobic respiration uses hydrogen gas as the electron donor and sulfate as the electron acceptor. And the waste product of this, I didn't show the waste product of hydrogen. This becomes protons, and the waste product of sulfate becomes hydrogen sulfide gas, which smells like rotten eggs, or other various things. So this is a very common thing in the environment, and many places smell like rotten eggs. So many places have this kind of anaerobic respiration going on, using sulfate as the terminal electron acceptor. And so if you've ever been to a sewer or smelled anything that came out of a human colon or been near an ocean, you might have smelled hydrogen sulfide because there's tons of sulfate in the environment. There are actually lots of microorganisms that use sulfur as an electron donor. So they use sulfur as their electron donors during aerobic respiration. So sulfur is their electron donor and oxygen is their electron acceptor, and that creates this sulfate as a waste product, which is then used in other environments as an electron acceptor. So that would be this. Sulfur as a electron donor, oxygen as the electron acceptor, and the waste product would be sulfate. Another very important form of anaerobic respiration is nitrate reduction, and that's where nitrate is used as an electron acceptor. And this is also called denitrification because the end product of this is nitrogen gas. And so nitrogen that might be in the soil or in a cell is lost as nitrogen gas, and it can't easily be brought back into the biological nitrogen cycle. It takes a lot of energy to convert this into anything a living cell can use. So denitrification is when nitrate is used as a electron acceptor. So we're going to look at one more form of energy generation, that's fermentation, and then we're going to put them all together and see kind of how they all work together. So fermentation is something you should be familiar with because muscle cells do it, and they do it when they can't get oxygen. So anytime there's no terminal electron acceptor, ATP can't be made using the electron transport chain or the proton gradient. So ATP must be made some other way. So if there's no terminal electron acceptor, like no oxygen, no nitrate, no sulfate, what can be done? How can a cell get ATP? Well, various pathways create some small amounts of ATP. So glycolysis generates 2 ATP every time glucose goes through it. Now remember I said something like the cell can get 25 or more ATP per glucose if it uses aerobic respiration. That's why our cells do it. That's why we use oxygen. We can get 10 times as much ATP for every molecule of glucose if we can send electrons through an electron transport chain. But if all we can do is send glucose through glycolysis, we get 2 ATPs for that. And this is a common thing in fermentation. Many different organisms can use glucose and they can use glycolysis, and they get the 2 ATPs from that. But here's the thing. Glucose is one of those pathways that takes electrons, Glycolysis is one of those pathways that takes electrons off of glucose. And that means it generates full electron carriers. So this is the full form of NAD. This is the empty form. So glycolysis requires this empty NAD plus and glucose. And at the end of it, it has a full NADH. That means, again, in glycolysis, the electrons are taken off of glucose and put onto this electron carrier, and it becomes this. Glycolysis can't happen without that. That's an essential part of glycolysis. So that means it is essential for the cell to remake this empty form of the electron carrier. So how does the cell make an empty form of an electron carrier if there's no external molecule to take the electrons? That's the central challenge of fermentation. They have to find a way to convert this full form of NADH back to the empty form. So they need somewhere for those excess electrons to go. And in fermentation, they don't have an external terminal electron acceptor like oxygen or sulfate or nitrate. So what do? They typically use pyruvic acid as their electron acceptor. Pyruvic acid is the final product of glycolysis. And so their different cells will do different chemical reactions with pyruvic acid, but all of them involve putting the excess electrons into pyruvic acid. So pyruvic acid acts as the electron acceptor. So the one I would expect you all to be most familiar with would be lactic acid, because that's what our own cells typically make as a waste product when they do fermentation. So if they don't have access to oxygen, they end up with excess electrons, and they quickly do glycolysis to get ATP, and that generates this product, pyruvic acid, and they put the excess electrons on the pyruvic acid. And the waste product of that is lactic acid. So this is also very common in bacteria. If you've ever eaten a fermented food like yogurt or kefir or kimchi or sauerkraut, you've tasted lactic acid. Lots of bacteria have evolved to make lactic acid as a form of chemical warfare. They can grow in the presence of lactic acid, but it's poisonous to their competitors. There are also a lot of bacteria that will make butyric or propionic acid, and these smell really, really bad. So propionic acid is what gives Swiss cheese its disgusting flavor. But high concentrations of it would make a person vomit. Smelling butyric acid in any high concentration would make anyone vomit. A bottle of butyric or propionic acid, they have a warning on them that says stench, and that's not kidding, because if you break a bottle of one of these things, you have to evacuate the building, because everyone who smells it vomits. You can't really work when you're vomiting. Anyway, they're disgusting, and lots of other fermentation products that we haven't really put here, slightly different processes, but always like excess electrons. What do cells do with them? They generate the molecules that give corpses and feces their distinctive odors. Because in corpses and in feces, fermentation is what's happening, because there just isn't any oxygen, and any other electron acceptors have been rapidly consumed. So the only thing left to do is fermentation. One thing I might ask you to think about is, like, how do you make sense of this slide? Which microbes are using which metabolism? Go ahead and pause this video, and if you have trouble with this, let me know. I'm going to keep going. So it's just a fact of life that anaerobic bacteria tend to smell bad, and they tend to correlate with things we don't like or which are hazardous to us, like feces and corpses. So the products of anaerobic respiration and fermentation, we don't ever smell them most of the time because oxygen is present most of the time, and anything that can use oxygen will use oxygen. But as soon as oxygen disappears from an environment, lots of other things start happening, like dental biofilms. And if you sleep with your mouth closed, there's no oxygen in your mouth. You wake up with bad smelling stuff in there, and that's fermentation products. Okay, so I was hoping to do this in one video, but I can't. So energetic competition is probably the coolest thing we're going to see here, and it will have to be in the next video.